friends, in this video I will explain how can you design a primer for a template DNA sequence, that DNA sequence you don't know. For example, in case of Sanger DNA sequencing or other kinds of DNA sequencing, you don't know the template DNA sequence, but you have to design a primer for that template for further replication or other processes. Okay, so for example, this process you have to clone your gene of interest into a vector. For example, uh, this black one is your vector gene segment. Here is the black one is the vector gene segment, and you have to insert your gene of interest within the vector. This is the gene of your interest, I indicates, and you design that vector, so they must have a restriction enzyme cutting site here, also a cutting site here, also a cutting site somewhere there. Okay, so for this process, first you have to denature the DNA segment. For example, here first you have to denature the DNA segment in this way, so to produce single strand in this way. So the denaturation step, denaturation single strand. Now you can use this single strand, this is single strand, this is your gene of interest. I will show you by just one strand, using one strand. Now you know the vector DNA sequence, right? So you can design primer according to that. So for example, now I can design a primer according to the vector DNA sequence. Here primer binds with the DNA, because primer is necessary. It's a 5' end and this is the 3' end. Now the 3' wedge, now the DNA polymers can use it and extend the DNA synthesis towards this way. Now I told you that wait a minute. I told you that this is the primer sequence, that there is a cutting site here, here and here. So you cut the DNA sequence from in this way, and now you can use this you can use this primer and the template DNA sequence for the sequencing process. Only this much is required, this much is required, this much is required. Now you can use these sequences as a template primer. Okay, that much you can use. Okay, now we can put them in tubes. Like in case of Sanger sequencing, there are 4 tubes. One is for A, one for C, T, G. Now you can use these primer sequences to each tube. You have to add, then you have to add DNTPs, also the DNTPs, which I will explain those things in later part of the video. Okay, next video. Thank you.